Good day, everyone. So today we'll be discussing about proteins, which is basically the continuation of amino acids and peptides. So the references for this lecture will be from Biochemistry by Campbell, 8th edition. So please read the chapter on the three-dimensional structure of proteins from Campbell. And I also got some information from Leninger's Principles of Biochemistry. So first we'll be discussing about the protein structure function relationship. So I've been emphasizing in the previous lecture about the importance of structure because it tells you a lot on its function. So knowing the protein structure will tell you the properties of a certain protein, on its characteristic, on its tendency to act on a specific function. So when we say proteins, proteins consist of polymers of amino acids linked by covalent peptide bonds. So from the previous lecture, the covalent peptide bond is also known as the amide bonds, and amino acids are known to be the building blocks of proteins. And we have the term native conformation. Okay, please take note, this is conformation, not configuration, because configuration tells you about more on the amino acids, the LND configuration. But when we deal about proteins, it's more on the conformation. Okay, when we say native, it refers to a structure of protein having a function, okay, or a biologic activity. So when we have the term native conformation, it refers to the three-dimensional shape of proteins having a biological activity. Whatever biological activity that is, as long as it's a functional protein, it refers to a native conformation. Okay, so a protein's conformation is largely stabilized by weak interaction. So we have the term stability. So this stability is the tendency to maintain, it's a tendency of a protein to maintain a native conformation. So again, what is a native conformation? This is the three-dimensional shape of proteins with biologic activity. So basically, stability is the property of a certain protein to maintain on its functional protein state or a functional structure. So a given polypeptide chain can theoretically assume countless conformation, and the reason for this is that a protein can have multiple alterations that can happen in terms of their amino acid structure, in terms of their side chain, the ionization of their side chain. Okay, so it can assume a countless conformation because of a lot of factors. Now, proteins in their functional folded conformation are what we call native proteins. So as long as a protein is functional, they are folded. When you say folded, and it is later on we will be discussing on folding of proteins. It refers to the state of a protein wherein there is hydrogen bonding between the side chains of a certain amino acid and hydrogen bonding of the adjacent amino acid or within that peptide chain. So when we say native proteins, they are just proteins in their functional state or a folded state. And there is a high degree of conformational entropy. So in a natural setting, there is a high degree of conformational entropy. And if you dissolve proteins in water or in proteins in a solvent, the tendency is that protein will have the tendency of a protein uh, placing it and it's on an aqueous environment. The tendency of that protein is to be on an unfolded state. Okay, so if there is an unfolded state, therefore there will be no function of proteins. So in order to counteract this one, there should be chemical interactions to stabilize that native conformation. So we have the following chemical interactions. We have disulfide, which are covalent bonds, the non-covalent interactions, the hydrogen bonding, hydrophobic interaction, and ionic interaction to counteract this entropy. Now remember again from the previous chapter in chapter 1 in biochemistry, when you say entropy, it is the state of randomness. It's the degree of randomness of a system. So if you put a protein in an aqueous environment, the tendency is that that protein will be favoring a state of randomness. And in order to counteract that randomness, we need to have these weak interactions. Now, proteins are organized into levels of structure. So we have primary structure of proteins, the secondary structure, tertiary structure, and quaternary structure. When we say primary structure, this is what we have been discussing on the previous lecture about the amino acid sequence of a peptide. So this is just the sequence of amino acid chains of a peptide. Okay, this is the one-dimensional structure of protein. It's the primary structure of protein. So, and the important bonding between two amino acids again are the covalent peptide or amide bonds. When we say secondary protein structure, this is the local folding of the polypeptide chain or a peptide chain into helices or sheets. So basically, this is the two-dimensional structure of proteins. When we have tertiary structure proteins, this is the three-dimensional folding pattern of a protein due to side chain interactions. And when we say quaternary structure of protein, this will now apply to those proteins having more than one peptide or the polypeptide chains. Okay, so we'll be discussing the individual organization of proteins, starting off with the primary protein structure. So the amino acid sequence greatly determines the three-dimensional structure of the protein. So if you have an amino acid sequence, okay, that amino acid sequence will tell you on its property, about the property or the three-dimensional shape of a protein. So for example, between fibrous and globular amino acid, the two of them have different amino acid compositions. So later on, you will be discussing about fibrous and the globular proteins, and there's a great variation on the amino acid composition because it is the primary structure of proteins that will tell you the overall shape, the overall function of a specific protein. And any alteration in one amino acid may greatly affect the biological function of the protein. Now, please take note, may greatly affect. It does not always mean that if you have an alteration of one amino acid, it can cause severe alteration in proteins because we have what we call silent mutations. And this is greatly exemplified with sickle cell anemia. Now, sickle cell anemia is a disease of the hemoglobin molecule. Okay, now hemoglobin is found inside the red blood cell or the erythrocyte. And this hemoglobin, the function of hemoglobin is to bind to oxygen. Number one is to bind to oxygen, and number two, to transport oxygen. So that is the main function of that hemoglobin. Now, what is the structure of hemoglobin? So the structure of hemoglobin consists of four polypeptide chain. Okay, we have four polypeptide chain. Two chains are identical. So we have identical alpha helices, okay, alpha chains, and identical beta chains. Okay, so in if we try to dissect the beta subunit of the hemoglobin, this is the normal primary structure of the beta subunit. So it consists of valine, histidine, leucine, threonine, proline, glutamate, and glutamate. Now, in the case of sickle cell anemia, the 6 amino acid residue is actually altered for valine. So it changed from glutamate to valine. Now, remember glutamate, this is a polar or hydrophilic amino acid. So it is more likely located on the outer surface of the protein or the beta subunit. Now, if there's a change to valine, this change in valine, now valine is a non-polar amino acid. So we have a change from a polar to non-polar amino acid, and this causes exposure of the hydrophobic region of the beta subunit. And this exposure of hydrophobic region of the beta subunit, it causes this sickle cell hemoglobin conformation. And what is important about sickle cell hemoglobin is that it changes the function of hemoglobin. Okay, again, what is the function of hemoglobin? It binds to oxygen and it transports oxygen. So if there's a change in this overall structure, this sickle cell hemoglobin has lesser tendency or has lesser capacity to bind to oxygen and to transport oxygen, and causes the sickling of red blood cells. Okay, so this is the uh, another illustration showing the importance of the amino acid sequence. So in the normal, this is the normal red blood cell, normal hemoglobin amino acid sequence. From this, the sixth amino acid in the beta subunit is glutamic acid. Now in sickle cell anemia, there's a mutation to a valine. Okay, instead of glutamic acid, the translated amino acid is valine. And this change in amino acid causes crystallization of the hemoglobin molecules, causing less binding to oxygen, causing the sickling of red blood cells. So that is sickle cell anemia. Okay. Now within each amino acid residues, there are two bonds with reasonable free rotation. Okay. So the bond between the alpha carbon and the amino nitrogen of that residue, and the bond between the alpha carbon and the carboxyl carbon of that residue, or the carbonyl carbon of that residue. Okay, so it tells you that in this one, if this is a peptide bond, this is a peptide sequence, and this is the peptide bond. Okay, there are two bonds with free rotation, meaning it can twist upon itself. And what are those bonds? What are those bonds? So these are the nitrogen of the amino group. Okay, for example, here the nitrogen of the amino group and the carbonyl carbon. Okay, what else? The carbonyl carbon and the it's the alpha carbon and the carbonyl carbon. Again, so the free rotation occurs between the bond of the nitrogen of the amino group and the alpha carbon, and the alpha carbon and the carbonyl carbon. So these two bonds have free rotation. Okay. Now what is important is that peptide bonds, which are the carbonyl carbon and the nitrogen of the amino group, this is rigid, meaning it has no ability to rotate, doesn't twist around. The only bonds that twist around are this one and this one, okay, this one and this one. So the nitrogen of the amino group and the alpha carbon, and the alpha carbon and the carbonyl carbon. Okay. So what is the reason why the peptide bond is rigid? Okay, there's a reason why there's a rigid peptide bond. It's because of the partial double bond characteristic of the peptide bond. So the oxygen has a partially negative charge, okay, and the hydrogen bonded to the nitrogen has a net partial positive charge, setting up a small electric dipole. Okay, and what happens that the six atoms of the peptide group lie in a single plane, with the oxygen atom of the carbonyl group trans to the hydrogen atom of the amide nitrogen. So because of the possible resonance of the electrons, okay, this imparts a partial double bond of the carbonyl carbon and the nitrogen of this entire peptide bond. Okay, so and this arrangement, okay, so this arrangement of the peptide bond, okay, it's very important for you to emphasize that the carbonyl group is trans to the hydrogen atom of the amide nitrogen. So when you say trans, it is opposite. So this is going up, and this is what is going down. So that is trans. When you say cis, it's on the same side. Okay. So this is trans because the carbonyl group, it is opposite to that direction of the hydrogen of the amide nitrogen. Okay, so this is the structure of the peptide bond, and this structure is rigid and planar. Okay, so please remember the peptide bond is rigid, and the reason why it is rigid, it's because of the resonance of the electrons. Okay, why is there resonance? It's because of the partial positivity of the nitrogen and the partial positivity of that oxygen. So there's a tendency of the electrons to resonate to one atom to another. Okay, so again, emphasizing the planar structure of the peptide bond. So the peptide bond is here. So this is the peptide bond. This is the carbonyl carbon and the nitrogen of an amide group. So this is rigid. Okay. So the carbonyl carbon and nitrogen, this comprise the peptide bond. Okay. As you can see, the oxygen bound to the carbonyl carbon, this is trans. Okay, trans to the hydrogen, meaning it has an opposite direction as that of the hydrogen bound to the nitrogen. And this forms a planar structure. Okay, it is a planar structure because this one forms the backbone. Okay, it forms a backbone. It is not movable. Okay, and what are the only bonds that are movable? It is the nitrogen of the amide and the alpha carbon. Okay, now whatever is attached to the alpha carbon, except for the R group, that is the one that is movable or that is rotational. Okay, so these findings concluded that the peptide bonds or the peptide carbonyl carbon and the nitrogen of the amide group cannot freely rotate because of their partial double bond character, and rotation is permitted around the nitrogen and the alpha carbon and the carbonyl carbon and the alpha carbon bonds. Okay, so I would like to emphasize again that rotation only occurs at the nitrogen and the alpha carbon and the alpha carbon and the carbonyl carbon. And no rotation happens between the peptide bond. So that is the carbonyl carbon and the amide nitrogen. This one, the carbonyl carbon and the amide nitrogen. This is not movable. It has no capacity to twist or to rotate. Okay, so the backbone of a polypeptide chain can thus be pictured as a series of rigid planes with consecutive planes sharing a common point rotation at C alpha or at the alpha carbon. The rigid peptide bonds limit the range of conformation possible for a polypeptide chains. So we have what we call the Ramachandran angles. So the Ramachandran angles are just the eponyms for the rotation of those bonds that has the capacity to rotate. So what are those bonds that has the capacity to rotate? These are the alpha carbon and the nitrogen of the amide, and the alpha carbon and the carbonyl carbon. So the name the name of the angle or the rotation between the alpha carbon and the amide nitrogen is what we call the phi angle, phi, while the rotation between the alpha carbon and the carbonyl carbon is what we call the psi. Again, the nitrogen to alpha carbon is what we call phi, while the alpha carbon to the carbonyl carbon is what we call psi. So the conformation of a protein backbone can be described by specifying values of the phi and psi for each residues, but we will not deal with those determining the values of the Ramachandran angles. Okay, so again, this is to emphasize that the peptide bonds, the one that you see here, they are not free to rotate. Okay, and it forms a planar structure. And why is there a planar structure? Again, it's because of the trans arrangement of the carbonyl group and the hydrogen of the amide group. So the oxygen is pointed upward. Here, the hydrogen is pointed downward. Here, the oxygen is pointed downward. Here, the hydrogen is pointed upward. So there's an opposite arrangement of the oxygen and the hydrogen. Okay, the oxygen of the carbonyl group and the hydrogen of the amide group. Okay, so that is the reason why there is a planar configuration, and that is not movable. This peptide chains are not movable. They have no capacity to rotate. Only those the phi and psi angles have the capacity to rotate. Okay, so please click this link later. Now this illustration and this animation shows you the rotation of the bonds in the peptide sequence or in a primary structure of protein. Now, just like to emphasize again and again, that rotation only occurs between the phi and the psi bonds. So again, the phi angle or the phi bonds or the phi angle or the phi rotation occurs between the alpha carbon, it is between the alpha carbon and the nitrogen, okay, and the alpha carbon and the carbonyl carbon, okay. So it is the one that rotates, and also emphasizing that the peptide bonds, which is the bond between the carbonyl carbon and the nitrogen, it is rigid. It has no rotation. And the reason why there's no rotation, it's because of the partial double bond character of the peptide bonds. So now let's move on to secondary structure of protein. So this is the secondary structure of protein. It deals more on the hydrogen bonding of the polypeptide chains. Now, the secondary structure of proteins is the hydrogen bonded arrangement of the backbone of a protein. And there we have two kinds of secondary structures that occur frequently in proteins. We have the alpha helices and the beta sheets. When we say alpha helices, these are rod-like structures of secondary structure of proteins and usually involve only one polypeptide chain. And we have beta pleated sheets. These are two-dimensional arrays of sheets. So they are flat-like structures that can involve one or more polypeptide chains. Now, this table shows you the difference between alpha and the beta structures. So when say alpha helices, their structure is more likely a coiled rod-like shape. For beta pleated sheets, they are fully extended sheet-like or flat-like structures. The axial distance between two amino acids for alpha helices, it's 1.5 Armstrong, while in beta pleated sheets, it's 3.5 Armstrong. The number of constituent polypeptide chain is usually 1 in alpha helices. For beta pleated sheets, it's 1 or more. So it can be 2 or more polypeptide chains. The hydrogen bonding between the nitrogen and the carbonyl groups in alpha helices occurs on the same polypeptide chain, while in the beta pleated sheet, it can be on a different polypeptide chain or within the same polypeptide chain. And the hydrogen bonding in alpha helices are parallel to the polypeptide backbone or parallel to the axis of the backbone, while the beta pleated sheet are usually perpendicular to the polypeptide backbone. So again, dissecting through the alpha helices is stabilized by hydrogen bonds parallel to the helix axis. So this is the helix axis, and hydrogen bonding occurs parallel to the axis of the polypeptide chain. Okay, so counting from the N-terminal end of the amino acid, so this is the N-terminal and this is the carboxyl terminal. The carbonyl group of each amino acid, so this one, the carbonyl group, it is hydrogen bonded to the amino group or the amide group of the amino acid 4 residues away from it. Okay, so to better illustrate that one, so this is the carbonyl group of one amino acid. Hydrogen bonding occurs 4 amino acids away from it. So 1, 2, 3, 4. The hydrogen of the amide group of that amino acid is hydrogen bonded to the carbonyl group 4 amino acid away from it. Okay, so similar to this one, this carbonyl group is hydrogen bonded to the hydrogen of the amide group 4 amino acids away from it. So 1, 2, 3, 4. Okay, so please remember that. Okay, so the helical conformation allows a linear arrangement of atoms involved in the hydrogen bonds, which gives the bonds maximum strength and thus makes the helical conformation very stable. And there are at least 3.6 residues for each turn of the helix, and the pitch of the helix, which corresponds to the linear distance between the corresponding points on successive terms, is 5.4 Armstrong. Okay, so every turn of the amino acids there are 3.6 residues. Okay, and the distance between each turn is around 5.4 Armstrongs. For the amino acid proline, it creates a bend in the backbone, causing because of its cyclic structure, and it cannot fit into the alpha helix because first, the rotation around the bond between the nitrogen and the alpha carbon is severely restricted, and second, the proline's alpha amino group cannot participate in interchain hydrogen bonding. So what is the reason why if you have proline, it causes bending? So remember from the structure of proline, the structure of proline is like this, okay? So it cannot form an alpha helix due to the lack of amide hydrogen, okay, and the presence of steric hindrance. So you have no available hydrogen for hydrogen bonding, and therefore you cannot create a hydrogen bonding for amino acids away from it. So that is why it causes bending. So other localizing factors or factors that will affect alpha helices. So um, this usually involves side chains having electrostatic repulsion owing to the proximity of several charge group of the same sign. So if you have side chains that contains the same charge, most likely it will cause repulsion. So for example, if you have lysine and arginine clumped together, it will cause repulsion or the repulsion of the side chains. It can cause alpha helix disruption. So but and also if you have glutamate and aspartic acid or aspartate, both of them have negative charges. The tendency is that they will be repelled, causing a change in the alpha helices. Now let's move on to beta pleated sheet. So in this beta pleated sheet, the peptide backbone is almost completely extended. And the hydrogen bonds can be, again, intra-chain or interchain. It can be within that polypeptide chain or with the other peptide chain. And we have two types of beta pleated sheet. It can be a parallel pleated sheet and anti-parallel pleated sheet. When you say parallel pleated sheet, the peptide chain runs in the same direction. And when you say anti-parallel pleated sheet, the alternating chains run in opposite direction. So we have two sheets, okay? For parallel pleated sheets, so this is the picture of parallel pleated sheets. This means that the directionality of the peptide chains run in opposite directions. For this one, this is the direction of the peptide chain from the N terminal to C terminal, and the other peptide chain is also from N terminal to C terminal. So they have the same direction. While in anti-parallel, they run in opposite. So for this one, the peptide chain runs from C to N, but for this one, it's N to C. So this is anti-parallel and this is parallel pleated sheet. This is parallel pleated sheet. Okay? So again, emphasizing the structure of the parallel and anti-parallel. For parallel, they have the same directionality. For anti-parallel, they run in opposite. Okay. So we have also supersecondary structures and domains. So the combination of alpha and beta strands is what we call the supersecondary structure. So if we have an alpha helix in combination with the beta sheet, that is considered a supersecondary structure. So we have different types of supersecondary structure. The most common one is the beta alpha beta unit. Okay, so this is a structure of the beta alpha beta unit. So we have the beta sheet. This is the alpha helix and the another beta sheet. Okay, so let me have a clear. Okay, so this is the beta sheet, the alpha helix, and beta sheet. So this is the most common supersecondary structure. So two parallel strands of beta sheets are connected by a stretch of alpha helix. So this is the structure of beta alpha beta. For alpha alpha unit, we have they also they we also call this as helix turn helix. It consists of two anti-parallel alpha helices. Okay, for this one there are no beta sheets. For a beta meander, it consists of an anti-parallel sheet formed by a series of tight reverse turns connecting stretches of polypeptide chains. So in beta meander, we have this like a zigzag configuration. So from N to C, Okay, and then this is N to C again, but the direction is opposite. So this is anti-parallel. This is also anti-parallel. Okay, so this is the beta meander. It's like a zigzag. Okay, for Greek key, this anti-parallel sheet is formed when the polypeptide chain doubles back on itself. So here in the structure of the Greek key, so this is the beta sheet. Okay, and then the direction is anti-parallel again with the first one, and then the next beta sheet is also anti-parallel to the previous one. Okay, and the next beta sheet is another anti-parallel structure against the previous one. So as you can see here, the configuration here is that the anti-parallel sheet folds back on itself. So that is the Greek key. We have also the term motif. When you say motif, these are repetitive supersecondary structure which tells us about the folding of proteins, but it does not allow us to predict about the biologic function because they are found in proteins and enzymes with very dissimilar function. Now, protein sequences that allow for a beta meander or Greek key can often be found arranged into a beta barrel in the tertiary structure of proteins. So these are the examples of motifs. So it can be beta meander or Greek key. They are usually arranged in a cylindrical shape, forming a beta barrel. Okay, so for example, this one in beta meander, again, the structure of beta meander is like this. This is usually arranged like in a barrel. Para siyang nasa cylindrical ang kanyang shape. It forms a hollow structure in the center. So, as what has emphasized, it's either the beta meander or the Greek key can form this beta barrel. Okay. Now, beta barrel can also occur with beta, alpha, beta units. And this arrangement occurs in triose phosphate isomerase from chicken muscle cells. Okay. So again, it forms a beta barrel structures. Now, collagen is the most abundant protein in vertebrates. So we have discussed about this one. I have introduced a little bit about collagen in the previous lecture on amino acid, particularly the glycine. Now, a collagen fiber consists of three polypeptide chain wrapped around each other. So this is the structure of collagen. It consists of three polypeptide chain that is wrapped around each other. And each of the three chains has a repeated sequence of three amino acid residues. So the usual amino acid residue the usual repeating sequence of collagen consists of X progly or X hydroxyproline or glycine. So this X is an amino acid which can be any amino acid, proline and glycine. Okay, X stands for any other amino acid. HYP stands for hydroxyproline and glycine. So as you can see, glycine constitute a major role in collagen. Now, proline and hydroxyproline can constitute up to 30% of the residues in collagen. And hydroxyproline is formed from proline by a specific hydroxylating enzyme after the amino acids are linked together. So proline is usually formed when hydroxyproline is usually formed when the amino acids have been formed, and an enzyme, a specialized enzyme, will cause hydroxylation of the proline residues. So in the amino acid sequence of collagen, every third position must be occupied by glycine. As you would notice here, the third position is glycine. You have glycine, 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 glycine. So every three amino acid, we have glycine. Okay. The triple helix is arranged so that every third residue or every glycine, the chain that glycine is within that helix. Okay. The purpose of the glycine is that so that glycine will be found in the inner part of the triple helix. Okay. So, and why is that glycine is located in this part of the triple helix because it has a small side chain. So it actually maximizes space for the triple helix, and it is small enough to fit into the space available. So it is the only amino acid that can fit the small space of the triple helix, the inside of the triple helix. That is why glycine is located in the inner part of glycine, of the collagen. Okay, so again, emphasizing glycine that occurs every three of amino acid in the collagen, and given this arrangement, glycine is located, okay, it is located inside the helix. It forms the center of the helix, and the reason why it is located at the center is because glycine has a small side chain, and it is small enough to fit the space available of the collagen, inside the collagen. Now, we have a question here. Why does meat from older animals is tougher than meat from younger animals? So, collagen is both intramolecularly and intermolecularly linked by covalent bonds formed by reaction of lysine and histidine residues. And the amount of cross-linking in tissues increases with age. So, consider this structure, okay, of collagen. So, as what I've previously mentioned, collagen consists of three polypeptide chains that are woven together to form this triple helix. Okay, that triple helix can form intermolecular covalent bonding, okay, or what we call cross-linking from one triple helix towards one another. They can form covalent cross-linking, okay, at one. Now, in younger collagen, the cross-linking are lesser. Compared to older collagen, the cross-linking are numerous. So we have more intermolecular bonding in older collagen, and because of this more intermolecular bonding or more cross-linking between lysine and histidine residues in older collagen, it makes this meat tougher compared to the meat of younger animals. So what is the role of vitamin C in collagen? So the role of vitamin C in collagen is that it is a cofactor in the hydroxylation of proline and lysine. So lysine and proline are usually hydroxylated once they are incorporated into the amino acids of the collagen. They are hydroxylated by this enzyme, proline and lysyl hydroxylases, and it requires ascorbic acid to function. Okay, without ascorbic acid, there will be no conversion of proline and lysine to hydroxyproline and hydroxylysine. So that is why in vitamin C deficiency, you have a problem in the collagen formation or in the collagen synthesis because you will not be hydroxylating any proline and lysine. So a condition called scurvy can result into bruising of the gums, bleeding of the gums because the collagen of our gums will be weaker, exposing the blood vessels of our gums, causing gum bleeding. So that is why we need collagen. We need vitamin C for collagen to be synthesized. So we have two types of protein conformation. We have fibrous and globular proteins. When you say fibrous proteins, these are proteins whose overall shape is that of a long, narrow rod, just like that of collagen. When we have globular proteins, these are proteins whose overall shape is more or less spherical. So like this one. So this is a structure of myoglobin. It's more likely spherical compared to a narrow, long, rod-shaped fibrous protein. So these are the differences of fibrous protein and globular protein. Now, fibrous protein consists of long parallel polypeptide chains forming helical structure or pleated sheets. They are usually insoluble in water because of this arrangement. The structure is more stable compared to the globular one, and it plays a role in mechanical and structural functions. So examples of fibrous proteins are keratin and collagen. And in globular proteins, these consist of coiled and folded polypeptide chains forming a spherical shape. They are usually soluble in water. The structure is more or what less stable compared to fibrous protein, and it takes part in metabolite and chemical processes. Examples of globular proteins are the enzymes and the globins, the myoglobin, and hemoglobin. Now let's move on to the tertiary structure of proteins. So we have different forces that are involved in the tertiary structure of proteins. Okay, so the forces that are involved in tertiary structure of proteins involve metal-ion coordination, hydrophobic interaction, disulfide bond, which is, by the way, the most common and the most important intermolecular force in the tertiary structure of proteins, hydrogen bonding, and as well as electrostatic attractions. So we have different forces. So we have hydrophobic, hydrophilic interaction, salt bridges, hydrogen bonding, and disulfide bonds. Okay, so disulfide bonds, they stabilize the native conformation of the protein. So as what I've previously mentioned, it is the most common and the most important intermolecular force for tertiary structure. So they are the ones, so these disulfide bonds are the covalent bonds that stabilize. Okay, we have the term stability. They play a role in the stability in maintaining the native conformation of the protein. We have also hydrophobic interaction. So in hydrophobic interaction, it's usually found in the interior of the protein to hide the hydrophobic residues. You have also hydrogen bonding formed between the polar residues and water, and between polar groups within proteins, and as well as ionic interactions. Okay. To exemplify the structure of an example of a tertiary protein structure, we have a protein called myoglobin. Now, myoglobin is a small protein found in the heart and skeletal muscles that bind to oxygen. So it is the first protein for which to complete the tertiary structure. So this is the first protein wherein its tertiary structure is recognized or is determined. And myoglobin consists of a single polypeptide chain, consists of 153 amino acid residues, and includes a prosthetic group called the heme group. Okay, so myoglobin consists of an 8 alpha helical regions, and there are no beta pleated sheets regions. So most of the secondary structure of myoglobin are alpha helices. And hydrogen bonding in the polypeptide backbone stabilizes the alpha helical region, as what we have previously mentioned. And polar residues or polar amino acids are located more on the outer part of or the exterior part of the myoglobin. It's because it is the only ones that has the capacity to form hydrogen bonding with water, and non-polar residues are the ones located in the interior of the molecule by having hydrophobic interaction. We have two polar histidine residues that are found in the interior of myoglobin, and these two polar histidine residues are involved in the interaction with the heme group and the bound oxygen. Okay, so this is the structure of the of the heme group. So as you can see, there are two histidines. So this is the histidine, the first histidine, and the second histidine. The first histidine is what we call the proximal histidine. It forms a coordination with the iron of the heme group. Okay, and we have also the distal histidine. Okay, and one molecule of myoglobin can bind to one oxygen molecule. So this is the usual binding site of oxygen. It binds to the iron of the heme group, and we have the histidine part in this region. So we have a distal histidine, and this histidine on the distal histidine is function for the steric hindrances for the binding of oxygen. So again, another illustration showing you the heme structure of myoglobin. So we have the proximal histidine. This is the heme group, and oxygen binds to the heme group. Okay, it doesn't bind to the histidine itself, um, but the purpose of this histidine is to form um, to form a hysteric hindrance in this part of the myoglobin molecule. Okay, the planar heme group fits into a hydrophobic pocket, and this is held in position by hydrophobic attraction between heme porphyrin ring and the nonpolar side chain of the proteins. So this is the whole structure of the heme. It is in this region where it is in this part, particularly the iron, where the oxygen is bound to. And these are the porphyrin rings. See here, these are the porphyrin rings, and these porphyrin rings is embedded to myoglobin by the virtue of hydrophobic attraction or hydrophobic interaction between the non-polar amino acids of myoglobin. Okay, so this the presence of the heme group affects also the conformation of the polypeptide. And when you say apoprotein, okay, this apoprotein refers to the polypeptide chain alone without the prosthetic heme group. If we remove the heme group from the myoglobin, that is what we call apoprotein. This apoprotein is not as tightly folded as the complete molecule. So this means that heme plays a role in the conformation of myoglobin. It's not just the composition of amino acid, but also the heme. We have the terms denaturation and folding. When you say denaturation, this is the unfolding of protein. That is the disruption of the tertiary structure. So in denaturation, you're actually disrupting the tertiary structure of proteins. And in denaturation, the non-covalent interactions that maintain the three-dimensional structure of a proteins are weak, and they are the ones that are disrupted. And we have several methods of denaturing proteins. We have heat, extremes in pH, detergents, adding oxidizing and reducing agents, and adding other substances. Okay, so in denaturation, what is important is that there's a disruption of the tertiary structure. And why is there a disruption of tertiary structure? Because the weak interactions are disrupted. Okay. So lastly is the quaternary structure of proteins. So the final level of protein structure pertains to proteins that consist of more than one polypeptide chain. Okay, so each chain is called a subunit. Okay, and the number of chains can range from two to more than a dozen, and the chains may be identical or different. The chains between the multiple polypeptide chains interact with each other non-covalently. Okay, the key term here is non-covalently via electrostatic attractions, hydrogen bonds, and hydrophobic interactions. So this is the structure of the quaternary protein structure. It consists of more than one polypeptide chain. In tertiary structure, we are only dealing with one polypeptide chain folding to folding on itself. But in quaternary, it consists of more than one polypeptide chain. So it can be a dimer, meaning it consists of two polypeptide chain. It can be homodimer. It means that both of them are identical to one another. You have also heterodimer, which means that the polypeptide chains are not identical or are different from each other. You have a trimer. So example of a trimer is the collagen. It consists of three polypeptide chains arranged into a helix. And we have also tetramer, example of which is hemoglobin, consisting of four polypeptide chain, but the two of them are identical or are homodimers. So the most concise or most significant example of quaternary structure of protein is hemoglobin. Now, hemoglobin is a tetramer consisting of four polypeptide chains. We have two identical alpha chains and two identical beta chains. Now, both alpha and beta chains of hemoglobin are very, very similar to myoglobin chain. And the alpha chain of the hemoglobin is 141 residues long, The beta chain is 146 residues long. And many of the amino acids of the alpha chain, the beta chain, and myoglobin are homologous, which means that they have the same amino acids in the same position. So that is why it's somewhat kind of related and homologous. Okay. For in hemoglobin, in contrast to that in myoglobin, one molecule of hemoglobin binds to four oxygen molecules. Okay. And what is unique about hemoglobin is that binding of oxygen to hemoglobin exhibits positive cooperativity. When you say positive cooperativity, this means that when one oxygen molecule is bound to hemoglobin, it becomes easier for the next oxygen molecule to bind to. So this is the structure of the hemoglobin. As you can see, it consists of four polypeptides, two identical alpha, and two identical beta. If one oxygen molecule binds to one subunit, which binds to alpha, by the way, each of these subunits has the capacity to bind to one oxygen molecule. That is why one molecule of oxygen can bind to four oxygen molecules. So if an oxygen is bound to alpha, this causes the next oxygen molecule to bind easily to the neighboring um, subunits of hemoglobin. So that is why it has this positive cooperativity, because binding of one oxygen causes an easier way for the next oxygen to bind to that hemoglobin molecule. Okay. So to compare hemoglobin and myoglobin, hemoglobin functions for oxygen transport while myoglobin functions for oxygen storage. Okay. Okay. Hemoglobin is for transport. Myoglobin is for storage. Hemoglobin is located in erythrocytes, while myoglobin is located in skeletal muscle as well as in cardiac muscle. Hemoglobin consists of four heme groups, okay, while myoglobin has one heme group. The hemoglobin contains two alpha chains and two beta chains. For the alpha chains, it consists of 141 amino acids. For the beta chains, it consists of 146 amino acids. But in myoglobin, it only consists of 153 amino acids all in all. Hemoglobin is a tetramer, meaning it consists of four polypeptides, two of which are identical to each other, while myoglobin is a monomer. Hemoglobin binds to four oxygen molecules. Why is that? It's because it is a tetramer. Each subunit of the hemoglobin, each of the four subunit of the hemoglobin can bind to one oxygen molecule. Therefore, it binds to four oxygen molecule. But in myoglobin, it only binds to one oxygen molecule. For hemoglobin, it exhibits positive cooperativity, while myoglobin does not exhibit positive cooperativity. The hemoglobin has a sigmoidal oxygen binding curve, while myoglobin has a hyperbolic oxygen binding curve. Okay, so if you'd like to compare the curve of hemoglobin and myoglobin has a hyperbolic curve, while hemoglobin has a sigmoidal curve. And what is the reason for this? Since you have only one available binding site for oxygen for myoglobin, okay, this means that uh, that myoglobin is already saturated by binding to one oxygen molecule. It has the highest oxygen affinity. Kasi 'di ba, isa lang yung binding niya. So, if magbabind siya, yun na yun. Puno na ang kanyang capacity to bind to oxygen. So, kahit anong increase of oxygen, okay? Kahit anong increase of oxygen level, the myoglobin is already saturated because it consists of only one binding site for oxygen. But in hemoglobin, since you have four available binding sites for hemoglobin for oxygen, if you increase the concentration of oxygen, okay, it causes if you increase the concentration of oxygen, the 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 saturation of hemoglobin is somewhat staggered. Okay, because binding of one oxygen molecule at one heme group, it increases the oxygen affinity of the remaining heme group in the same hemoglobin molecule. Kasi may available ka pa na mga binding sites for oxygen. So that is why it is more of a sigmoidal. Kasi sa myoglobin, diretso na, puno na agad yung binding sites mo. But in hemoglobin, you still have available sites for binding to oxygen. That is why it's more of a sigmoidal curve. Okay? We have also conformational changes that accompany hemoglobin function. So both hydrogen and carbon dioxide, which themselves bind to hemoglobin, affect the affinity of hemoglobin for oxygen by altering the protein's three-dimensional structure. So we have what we call Bohr effect. So this Bohr effect, it is the effect of hydrogen ions in the oxygen binding of hemoglobin. So if you increase the concentration of hydrogen ion, therefore lowering the pH, it causes the reduced affinity of hemoglobin. And what is the reason for this? If you increase the pH, why is the oxygen um, why is the oxygen affinity of hemoglobin is low? Because if you increase the hydrogen ion concentration, or if you decrease the pH, this causes the protonation of key amino acids. Okay, remember from amino acid that it can be protonated at low pH, including the end terminals of the alpha chains and histidine 146 of beta chain. This causes the protonation of histidine, and this protonated histidine is attracted to and stabilized by a salt bridge to aspartate 94, favoring the deoxygenated form of hemoglobin. So it basically changes the conformation of the hemoglobin. And this conformation of the hemoglobin is not has lower affinity to oxygen uh, compared to the hemoglobin at a normal pH. So that is why at the lower pH, or a concentration where there's high hydrogen, the binding of oxygen is lower. Therefore, it easily gives off oxygen. That is why in regions of our body where there is low pH, for example, in exercising muscle cells, the pH concentration at that region is usually low. Now it causes the hemoglobin, if a hemoglobin molecule passes through that region, oxygen is easily released from the hemoglobin because of the deoxygenated form of hemoglobin is favored, or the affinity of that hemoglobin to oxygen is lower. So there will be easier release of oxygen at that region. Okay, so this is a summary of Bohr effect. So this table shows you the effect of pH in the several parts of our body. So, for example, in the lungs and the actively metabolizing muscle. So, in the lungs, you have a higher pH than actively metabolizing tissue. So, for example, if you are performing exercise, the muscle is very active. It's very active in terms of metabolism. And in that muscle, you have low pH due to production of acids. And aside from production of acids, you have also lactic acid that promotes decrease in pH. Okay, so this is the setup. So in lungs, there's a higher pH than actively metabolizing tissue. In the actively metabolizing tissue, or in actively metabolizing muscle, the pH is lower. It's more acidic because of the production of hydrogen. So in the lungs, hemoglobin binds to oxygen easily, while in the actively metabolizing muscle, hemoglobin releases oxygen. And why is that? It's because hemoglobin binds to hydrogen. And what will happen if hemoglobin binds to hydrogen? Again, it causes this process. It favors the deoxygenated form of hemoglobin. Basically, it changes the conformation of the hemoglobin that will not favor binding to oxygen. Therefore, hemoglobin can easily release its oxygen. to the actively metabolizing muscle. But here in the lungs, hemoglobin binds easily to oxygen, okay, because you don't have this factor. Yeah, because you don't have that hydrogen, you don't have that low pH or that increase in hydrogen concentration uh, causing this process to occur. So that is why in actively metabolizing muscle, hemoglobin easily releases its oxygen. I think it also makes sense na in active metabolizing muscle, the hemoglobin should easily give off oxygen to provide to provide oxygen for the actively metabolizing muscle, because in actively metabolizing muscle you need to have a lot of oxygen, okay, for the electron transport chain to occur. Okay, so that is the summary of the Bohr effect. So please read in the book for more details and information about the Bohr effect. And this ends my lecture on proteins. Please, please read the chapter of the three-dimensional structure of proteins, because it's very impossible for me to cover the entire contents of the book, and it really helps you in understanding the the functions and the properties of proteins if you have a good and a solid foundation and understanding of the three-dimensional structure of proteins. Okay, so thank you everyone and stay safe always.